Great. All right, just give folks a second to come in. Great. Well, welcome everyone. Today's speaker is Patrick Brunner. Dr. Brunner is an associate professor of dermatology at the Medical University of Vienna in Austria. He's a physician scientist and cares for patients suffering from chronic inflammatory skin diseases, as well as cutaneous lymphomas. Dr. Brunner completed his dermatology and dermatology residency at the Medical University of Vienna. He subsequently trained with Emma Gutman and James Kruger at Rockefeller, where he had a number of important contributions. He's really been a leader in applying newer technologies to inflammatory skin diseases and dermatology, and we're fascinated to hear more about his work today. And so I'm very excited to have you today, Dr. Brunner. Thank you so much for joining us. I'll turn it over to you.
Thank you so much for the kind introduction and for the invitation to talk about some things that we did in atopic dermatitis. Let me just share my screen. So you see my screen and the arrow?
Yeah, everything looks great. I think we're good to go. Thank you.
So thanks so much for the invitation, and I'm excited to talk about some aspect of the skin disease memory that we recently got interested in. So these are my disclosures. So atopic dermatitis is the most common chronic inflammatory skin disease. It typically starts very early in life. In a subset of patients, this disease can develop a lifelong chronic disease, while in other patients or in a other subset of patients, the disease goes away during adolescence or childhood. Adding to the complexity of atopic dermatitis, and despite the fact that this is the most common chronic inflammatory skin disease, it's a disease that does not even have a common name. So some call it eczema, some call it atopic dermatitis, some call it neurodermitis, but it all refers to the same disease or the same spectrum of diseases actually. And there are two phenomena in atopic dermatitis that I found particularly interesting. So first, in many patients, atopic dermatitis resolves during childhood and adolescence, but in other subset it can come back later. And in another subset, the disease develops for the whole life. And it's not really clear why it disappears in one subset and it doesn't disappear in the other. Or put it differently, why there's some disease memory in one patient, not in the other. And importantly, when you treat chronic atopic dermatitis, especially in adults, you can treat it now with different treatment modalities, but usually the disease comes back after cessation of treatment. So there needs to be some tissue resident memory that you cannot get rid of with current treatment modalities. So before I want to go into that aspect, I just want to highlight some concepts of atopic dermatitis pathophysiology. So it's you will often read that atopic dermatitis is a mixture of genetic and environmental factors, and they contribute to immune and epidermal barrier dysfunction. But the exact interplay is not entirely clear yet. So there are two major concepts in atopic dermatitis, the inside out and the outside in concept. So the outside in concept says there is a primary defect in the epidermal barrier, and a defective epidermal barrier allows pathogens or allergens to penetrate the skin and then elicit some sort of immune response. And there's the other dogma, if you want, or if there's another dogma of the outside in, of the inside out hypothesis, where you assume a primary immune dysfunction that then elicits inflammation in the skin, which then secondarily damages the epidermal barrier and leads to the atopic dermatitis phenotype. So those distinguishes are a bit artificial, and both concepts most likely hold true in atopic dermatitis. So why the barrier? So when you look at children very early on in life, and you measure their epidermal barrier via transepidermal water loss as a surrogate marker for the epidermal barrier, you can stratify those children in those that have a low transepidermal water loss, which means they have a better barrier, and high transepidermal water loss children which have an impaired barrier. And you see that the probability to stay without atopic dermatitis decreases more steeply when you have a bad barrier, which means when you have high transepidermal water loss. So when your barrier is not fully, or is presumably not fully functional. And one aspect to that barrier component is Filagrin, and I'm sure you heard a lot about it already. So Filagrin is an important component that seals off the epidermis, and there is a loss of function mutation in a subset of atopic dermatitis patients, and those patients have an impaired epidermal barrier. So loss of function of this Filagrin that explains development of atopic dermatitis in a subset, but not in all patients. And it's actually that worldwide, the majority of patients do not have this Filagrin mutation. But importantly, Filagrin can also be downregulated by atopic dermatitis inflammation. So, but this, of course, is a secondary phenomenon, not the primary loss of function. The situation with Filagrin decreases is actually a bit more complicated than initially thought, because when we looked at children versus adults and looked at Filagrin expression levels in children and in, and in adults, you see that you get this decrease in Filagrin expression in adults due to downregulation in AD inflammation. But this downregulation was not, we didn't find this downregulation actually in children. So while this Filagrin barrier downregulation seems to be more important in adults, it might not fully hold true in children. When we looked at neutral lipid deposition in pediatric and in adult AD, and when you look at healthy control skin, you see this red layer of staining. So these are actually neutral lipids that you can stain with Nile red, and in healthy pediatric and in healthy adult skin, you see nice deposition of these lipids, but these lipids are actually gone in adults as well as in pediatric atopic dermatitis, both lesional and non-lesional skin. So a defective lipid deposition might contribute, especially in children, to this impaired epidermal barrier. So there was then the concept, if you aggressively put emollients on children very early on in life, you might be able to prevent atopic dermatitis development in these children. And there were some initial promising results, but the larger study actually showed that there was not much difference between intervention and control populations. So restoring the epidermal barrier with emollients very early on in life is, of course, an intriguing concept, but right now the data does not really support that it really can prevent the advent of atopic dermatitis development. So this was sort of the barrier side. The inflammatory side in atopic dermatitis is, of course, very obvious because it's a chronic inflammatory skin disease. And there's one arm of the immune system that seems to be centrally involved in atopic dermatitis development, which is actually the Th2 immune axis. And this Th2 immune axis is characterized by the production of certain Th2-associated cytokines, such as interleukin-4 and interleukin-13. And this inflammatory arm of the immune system really seems to be very central to atopic dermatitis development, which was essentially proven by the clinical efficacy of Dupilumab, which is a monoclonal antibody that blocks interleukin-4 and interleukin-13 signaling via blocking the interleukin-4 receptor. And when you look at those phase 3 studies, for example, those are changes in clinical scores from baseline. Lot of or strongly decreased clinical disease activity in patients that receive Dupilumab as compared to the placebo group. And it's an approved medication and, of course, an established treatment in atopic dermatitis. And while there is considerable heterogeneity in atopic dermatitis between certain population and also between ethnic groups and between adults and children, the Th2 immune axis seems to be central to inflammation in all of these populations. Interestingly enough, it's not really clear what's the physiologic or side of a Th2 inflammation. I mean, Th1 inflammation is important for fighting cancer or tuberculosis, and Th17 for bacterial defense and to fight candida and regulatory T cells in order to dampen immune responses. Th2, the Th2 immune axis has always been assumed to be important for fighting parasites, worms, but in our, or at least in our Western lifestyle, it seems that this arm of the immune axis has lost somehow its role in protective immunity, and is actually mostly found in atopic diseases, including atopic dermatitis. So given this inflammatory component of atopic dermatitis, there's one very interesting phenomenon, which is that not only lesional, but also non-lesional atopic dermatitis skin, so skin that looks clinically normal, is also, has also some increased levels of inflammation, including Th2-associated inflammation, especially in patients with moderate to severe disease. And Emma Gutman has very nicely shown it, that in non-lesional atopic dermatitis, there are increased levels of interleukin-13, for example, when you compare it to normal disease. And there have also been papers showing that there's a residual disease profile also in treated atopic dermatitis. So when you treat atopic dermatitis, it never really goes back to normal skin. There's still some residual inflammation going on, and this is what that's what we became interested in. Because this might constitute somehow some sort of immune disease or some sort of disease memory in atopic dermatitis. And Liv Eidsmo from Sweden actually nicely showed in psoriasis that you get tissue-resident memory T cells sitting in the skin that you can't get rid of with a treatment. So she looked at interleukin-22 and interleukin-17 producing T cells, tissue-resident T cells in the skin that you find in active psoriasis, of course, but also in treated and long-term treated psoriasis. There were still several populations that could produce a lot of interleukin-17, interleukin-22 upon ex vivo stimulation that was not present in healthy control skin. So even during long-term treatment, there's something sitting in the skin, tissue-resident memory cells in psoriasis that might mediate relapses in those patients. And it's actually also known from allogeneic stem cell transplantation that certain tissue-resident memory cells you can't even get rid of with aggressive radiation and chemotherapy and allogeneic transplantation. So those cells tend to sit very firmly in the skin. So we wanted to have a closer look at atopic dermatitis skin, what's going on in atopic dermatitis skin after short and long-term treatment with the interleukin-4 receptor blocker Dupilumab, for example. Because when you stop this treatment, the disease usually comes back. And atopic dermatitis patients were not so thrilled giving repeated skin biopsies, and this is why we thought about an alternative sampling method. There are several papers out now that show that, for example, tape stripping is a very elegant and minimally invasive way to sample skin, both mRNA and protein from the skin, and you can also do disease monitoring with that. The advantage is, of course, it's easily done and you don't need to perform biopsies in those patients, but of course, you're not going to get cells out of the skin. So this is why we thought about the different sampling method that has been actually around for quite some time, and this is suction blistering. So suction blistering works in a way that you have a negative pressure chamber on your skin, and on the bottom plate you get holes, and via this negative pressure blisters form over the course of 1 to 2 hours, and you can have these plates custom made. We took this 5 millimeter 5-hole plate and put it on the skin. And for example, there have been studies already investigating this in inflammatory skin diseases such as vitiligo. And the idea is that you have this pump that puts some negative pressure on those chambers, and over the course of 1 to 2 hours, a blister develops. And in this blister, you do not only have interstitial fluid, but you also have cells migrating into this blister fluid that you can then harvest and profile using flow cytometry or single-cell RNA sequencing, for example. So this is an atopic dermatitis patients where you get 5 blisters, and you can puncture those blisters and get the interstitial fluid and cells out, but you can also harvest the blister roof, which essentially is the epidermis, which you then quickly can quickly digest it with trypsin, and then also perform flow cytometry or single-cell RNA sequencing with this method. Here's the epidermal sheet that you get, and both in the suction blister fluid and, or not only in the epidermal sheet, but also in the suction blister fluid, you get many cells that you can then investigate. For example, here T cells, memory T cells, both CD4 and CD8. And from the epidermal sheet, of course, you get lots of keratinocytes, but also leukocytes, Langerhans cells, and T cells mainly. And you also get a lot of melanocytes, actually, especially in the suction blister fluid. What we wanted to investigate was at this point whether suction blistering is or can be as good as a biopsy, a sampling, at least for certain cell populations. So we compared suction blistering and skin biopsies. We enriched for CD45 positive cells and then performed single-cell RNA sequencing. And in addition to the cells, we of course got the interstitial fluid out of the suction blister cells, and we analyzed those with a proteomic multiplex assay, which I will explain you in a minute. So when we then compared biopsy sampling in atopic dermatitis with suction blistering, we essentially found that most cell populations were actually present both in also in the suction blister population. Keratinocytes, actually all T cell populations, most dendritic cell populations, and actually melanocytes. If you're interested in fibroblasts or smooth muscle cells or endothelial cells, of course, suction blistering is not the method of choice, but for most cells, it works actually very well. Mast cells actually were not there, and also some populations of macrophages were not present in suction blistering, most likely because those are very sessile cells. If you think of tattoos, for example, that is taken, or tattoo dye that is taken up by macrophages, those cells really don't leave the skin and they don't migrate. So those populations are not really present, but other populations were actually consistently found in suction blistering. We then performed single-cell RNA sequencing because it's a great method also to handle small amounts of cells, and we compared biopsy and blister resolution. The resolution was good both in biopsy and blister sampling, but we found actually one thing that was very interesting. For example, keratin 6A, supposed to be present in keratinocytes, was present in keratinocytes, but there was also low-level signals in dendritic cells or T cells where these keratinocytes actually should not be present. We found it in biopsy samples, but not so much, actually not in blister samples. And we think that the method of suction blistering is more gentle in getting those cells out, and you don't need to have aggressive enzymatic digestion from biopsies where you might get more ambient mRNA contamination. So the resolution, the quality of the cells was actually better in suction blistering than in the biopsy samples. But still you get the disease signature that you would expect in atopic dermatitis. So you would expect Th2 inflammation. The lead cytokine is interleukin 13, and we found it in T cells, both of biopsy and suction blister samples at higher levels or higher frequencies than in healthy controls. And we also found interleukin 22 and also a little bit of interleukin 5, both in biopsy and blister samples. So here comparable profiles in T cell cytokine expression comparing blister and biopsy samples. The advantage of suction blistering is that you also get interstitial fluid, and it's not just serum that goes in. There are several papers showing that you get really interstitial fluid out of the skin when you compare it to serum. And I mean, the amount that you get out is very little. So from one patient, if you do the antecubital fossa, that's the location where we performed suction blistering, you get like 50 or 100 or 150 microliters of interstitial fluid out of from one patient. Of course, you cannot perform ELISAs with that amount, but there's now, for example, this Olink proteomic multiplex assay where you only need a few microliters of sample in order to profile several hundreds of markers. And it works via an antibody pair that is bound to a synthetic DNA strand, which then hybridizes, and then you can actually read a quantitative PCR and then calculate back the amount of protein that is present in the sample. And we found consistent upregulation of several pro-inflammatory mediators. And there was a lot of heterogeneity in atopic dermatitis samples, but this is to be expected in this population. But there's more yellow and red in the atopic dermatitis population compared to healthy controls. And there were several markers like CCL13 upregulated in atopic dermatitis blister fluid compared to healthy controls. And concordance of Olink proteomic assays was actually quite good, with levels found in single-cell RNA sequencing, like in the upper right or lower left quadrant. So now with this method, we asked the question, is it possible to find certain cellular or molecular components in patients that are treated with Dupilumab that are in clinical remission for 16 weeks or 1 year, whether we can find differences from healthy control skin. So we profiled atopic dermatitis, untreated atopic dermatitis after 16 weeks of Dupilumab treatment and after 1 year of Dupilumab treatment. And this was all performed in the antecubital fossa. And this skin was free of clinically visible disease. And when we compared cell epidermal thickness, it turned back to normal. When we looked at T cell counts after 1 year of Dupilumab treatment, it returned back to normal. And when we looked at, for example, dendritic cells, those numbers returned back to normal. So from a clinical and histopathological standpoint, these 1 year treated Dupilumab patients, they all looked normal. But when we took a deeper look into the transcriptomic makeup and also in the cell populations, we found some differences from healthy control skin. So when we looked at dendritic cells, there was one small population, we call it DC2 here, that was LAMP3 positive, that actually was there at increased frequencies even after 1 year of Dupilumab treatment compared to healthy controls. So of course, this is a relative distribution and not absolute distribution, because absolute cell numbers go down. But absolute cell numbers are very much comparable between 1 year of Dupilumab and healthy controls. So here you can compare and you see that there's a higher proportion of these DC2 cells compared to healthy controls, which were virtually absent in healthy control samples. As you can see here in this histopathological slide. So LAMP3 positive cells were present in baseline atopic dermatitis and in 1 year Dupilumab treated patients, but we did not find it consistently in healthy control patients. And those cells produced the Th2-associated chemokines CCL17 and CCL22 even after 16 weeks or 1 year of Dupilumab treatment. This signal was not present or present at substantially lower levels in healthy control tissue. So a cell population that you can't get rid of, a dendritic cell population that still is there after 1 year of Dupilumab treatment, that is not there in healthy control patients. And it seems that those cells produce also interleukin-15, which is actually a T cell growth factor. And you could even not only detect it in those cells, but when we used a confirmatory population, an independent validation group of bulk mRNA isolation from atopic dermatitis patients treated for 1 year with Dupilumab, we found actually increased levels of interleukin-15 in these treated populations. So when we did not only, of course, look at the dendritic cell population, but also at the T cell population. And this is, of course, a very interesting population because tissue resident memory T cells are assumed to play important roles in tissue resident memory. And we found several populations of T cells to be present in our samples. And when we looked at certain T helper cell lead cytokines, such as interleukin-13 for Th2, interleukin-26 for Th17, or interleukin-22 for Th17, Th22, There were there were increased frequencies of cells expressing these pro-inflammatory cytokines, not only at an untreated AD, but also after 16 weeks of Dupilumab and even after 1 year of Dupilumab. And this signal was not there in healthy control, in age and location matched healthy control samples. So there was still some smoldering inflammation going on despite clinical clearance, which was not seen for interferon gamma, for example. And when you calculate frequencies or not frequencies, but percentages of cells within the T cell pool, you see elevated levels of interleukin-13 positive, CD3 positive cells, both of the CD4 and CD8 compartment. Also some increase in interleukin-26 and interleukin-22. So those cells are maintained even after 1 year of Dupilumab treatment. And there was one subset that was especially interesting, this TC3 subset, because this subset was absent in healthy controls and only present in AD, but maintained through a Dupilumab treatment. And this TC3 population actually was positive for CRTH2, a marker that was actually previously described in a subset of Th2 cells, so-called Th2A cells that are found in allergic patients or patients allergic to environmental and food allergens. And when you look at our samples here at baseline, 16 weeks and 1 year of Dupilumab treatment, you see this population of, so PTGDR2 is actually the same as CRTH2. Just the G name, you see that this cell population persists throughout treatment, even after 1 year of Dupilumab treatment. It's totally absent in healthy controlled populations. And there's one, we could also corroborate this in histopathological stainings. So here in healthy controls, CRTH2 positive cells are not present, but they are there in atopic dermatitis, untreated atopic dermatitis patient skin, as well as after 1 year of Dupilumab treatment. And this cell population has an interesting receptor profile. They express the TSLP receptor, they express IL-17RB and IL-1RL1. And those receptors are the receptors of epidermis-derived alarmins, which are thought to be important in atopic dermatitis development, interleukin-22, interleukin-33, and TSLP. So those cells are sitting there in treated atopic dermatitis skin, and they might quickly react to epidermis-derived stimuli because they have all the receptors for these epidermis-derived alarmins. And as I mentioned, this population of Th2 cells that were called Th2A in this population by Wombre et al., they are actually have been previously described in allergic patients. And these cell populations have been described to decrease with successful peanut-specific treatment, immunotherapy. So also on a function level, they might be, or they are of course an interesting candidate for not only allergy treatment, but they might sit in the skin and mediate, for example, flare-up of the skin due to environmental allergens, and might be key cells mediating this skin resident disease memory potentially. We also did not only look at single-cell RNA sequencing at the mRNA level, but of course we wanted to see whether this sort of smoldering inflammation holds true also on a protein level, because only because an mRNA is there doesn't mean that the protein is also expressed. So here we compared untreated AD with healthy control suction blister fluid and performed this multiplex proteomic assay. And you see a lot of pro-inflammatory mediators are upregulated, also CCL17, which is an important atopic dermatitis activity marker. And after 16 weeks of Dupilumab treatment, many of those inflammatory mediators actually go down, but suddenly interleukin-13 stands out. And here, after 1 year of Dupilumab treatment, when you compare it to healthy control skin, you see that there's one single cytokine in our panel that was still significantly upregulated. That is interleukin-13. So interleukin-13 on a protein level is still there even after 1 year of Dupilumab treatment. We also then looked at the peripheral blood in a cohort that we had available, and also performed these multiplex assays, and also looked at baseline 16 weeks and 1 year of Dupilumab treated patients. And again, interleukin-13 levels are maintained. Overall disease activity goes down as reflected by CCL17, as I mentioned, a very important disease activity marker. But interleukin-13 was still elevated above healthy control levels, even in the serum of these patients, suggesting this smoldering inflammation is also visible in the serum of those patients. So suction blistering actually worked very well for us in those patients, especially because they could get suction blistering repeatedly, and there's no scarring involved, and it's a relatively painless method. We could comprehensively characterize many potentially pathogenic cellular and molecular components of the skin. And it even works in treated skin where you only have little cell, or where you expect to have only little inflammatory cells to be present. And with this method, we actually identified these populations of persisting immune cells, dendritic cells and helper T cells, that might be suggestive of a skin resident treatment resistant immune memory. We also found the CD8 positive, CRTM positive population, but I didn't mention them here for time reasons. So there's this skin disease memory that you can't get rid of after treatment. What about those patients that spontaneously clear their disease? Because when you look at this plot that, that shows you the atopic march, a concept, of course, that doesn't hold fully true today, but still shows that atopic dermatitis and food allergy is mostly present in young children and less in adults. So why is it that children, or many children, lose their atopic dermatitis over time? One reason might be that there's an increase in sort of pro-allergic cytokine expression in not only in atopic dermatitis children, but also in healthy children. For example, interleukin-33 levels are increased very early in life compared to adults, and interleukin-33 has been shown also in animal models to be very important for food anaphylaxis via cutaneous sensitization. So there might be a different cytokine level that then naturally decreases over time. There's one very interesting study, not in skin, but in lung, atopic lung, or generally in lung development, that looked at factors that might elicit a more Th2 biased inflammation. And interestingly, Th2 cells have dopamine receptors, and dopamine or dopaminergic innervation can skew the immune environment towards some more Th2 skewed inflammatory or Th2 inflammation. And interestingly enough, at least in mice, when they grow up, dopaminergic innervation is decreased, and adrenergic innervation increases. And this might be a reason why very early in life atopic diseases are increased, like allergic asthma, as opposed to adult patients. But this is, of course, a pure speculation and needs to be corroborated. But there are these quite interesting results in asthma and in lung. We actually were interested in what, or given our suction blistering method, we investigated patients that spontaneously lost their atopic dermatitis, that still showed some sensitization to environmental allergens, increased IgE levels. But we looked at 4 patients, or 4 individuals that lost their atopic dermatitis during adolescence, and compared it to healthy control skin of individuals that never had any allergic sensitization or any atopic disease. And when you compare cell populations with the suction blister method in spontaneously healed AD with healthy controls, you see that the cell populations themselves are not differing. So we did not find any different cell population, at least on this single cell level, comparing spontaneously healed AD with healthy control with healthy control skin. We then looked at differentially expressed genes between spontaneously healed patients and healthy control patients, and plotted it here in this feature plot. and red color just means that there's some sort of there are increased numbers of differentially expressed genes within this cluster. I mean, the differences they're very small, and you might criticize, of course, that the differences are very, very tiny, but still there were some differences in some T cell subsets, and there were also some differences in melanocytes, for example, in Pla2G7, which actually degrades platelet activating factor, which is a strongly pro-inflammatory mediator. We don't really know what it means, but there seems to be a difference in melanocyte expression levels in spontaneously healed patients versus healthy control patients. When we looked at the T cell compartment, we actually found quite anonymous increases in TGF-beta levels in many T cell compartments in spontaneously healed AD, which is you could say a tolerizing inducing cytokine, or involved in some tolerizing mechanism. Of course, the immune effects of TGF-beta is very complex, but at least we found increases in TGF-beta in several T cell populations. Also, interferon gamma dependent signaling seemed to be somewhat increased, and there was also an increase in interleukin 27 receptor, interleukin 27, and anti-inflammatory cytokine. You could, of course, argue that this might only be a batch effect and differences are very small, but from a functional standpoint, we actually found quite consistently that especially regulatory and Th1 associated markers were increased in spontaneously healed AD, and this might indicate that these markers might be involved in spontaneous clearing of AD, and those markers were there at increased levels compared to healthy control patients. And we also looked at interstitial fluid, and when we compared spontaneously healed AD versus active AD, for example, one marker stood out, that was PSG1. And PSG1 actually is pregnancy-specific beta-1 glycoprotein 1, which is increased during pregnancy and assumed to protect the fetus from the attack of the maternal immune system. So this is sort of an immunosuppressive, immunomodulatory marker, and future studies will, or time will tell whether this marker might be involved in some tolerizing mechanisms in patients that spontaneously cleared from their atopic dermatitis. So in order to sum up, in spontaneously resolved AD, we found conventional T cell to show increases in certain regulatory markers, in the general skewing towards a more Th1-like phenotype. And this is also what you see very early in life. So when you look at blood, for example, in patients that later on develop atopic dermatitis, they have a more Th2 biased immune environment, whereas Th1 seems to be more protective. So this whole yin-yang seems, or is consistent actually with what you see early in life. Interestingly enough, we didn't find any differences in regulatory T cell expression or numbers, and also keratinocytes transcriptomics were essentially indistinguishable from healthy control skin. So with this slide, I would like to conclude. I would like to thank all the many people that were involved in these studies, especially my mentors Emma Gutmann, Jim Krüger, and Georg Stingel, and of course my funders, especially the Leo Foundation, who funded these studies, and also the Austrian Science Fund and the City of Vienna. And I'm very happy to take questions. Thanks so much for having me.
That was great. Well, thank you so much for that amazing talk. Really, really great work. So while we wait for some questions to come in here, maybe I'll start with one, if that's okay. So I noticed that IL-13 is the predominant type 2 cytokine in AD. Does IL-4 play any role?
So IL-4 is there also on a protein level, but it's there at much, much lower levels, both on an mRNA and also on a protein level. So IL-13 is there at a much higher order of magnitude.
Yeah, interesting. Thanks. We have a question. What do you make of the increased IL-15 expression in DCs after 1 year of Dupilumab treatment?
I mean, we did not only find it in those cells, but also in bulk isolates. So there's a signal also in independent samples. I mean, it's related to interleukin-2. It might, foster some, impact on tissue-resident memory cells and orchestrate their survival, maybe. I mean, but that's of course pure speculation, and we don't have any functional data to support this. But it's of course interesting to have like this survival factor there being produced by a population of dendritic cells that is Th2 biased and makes cells survive in the skin.
Interesting. I may ask another. So what do you think T cells are targeting the AD? Is it self-peptides or bacterial peptides? I realize that it may not be known, but I'm just curious what your thoughts are.
That's a tough question, of course. The question is whether they target anything or whether it's only some sort of bystander information.
Yeah, or anything else. Yeah.
Whether it's allergen or antigen driven at all. I mean, Thomas Vegf very, very elegantly showed in his pollen chamber that when you put extrinsic AD patients in a chamber and you blow pollen on them, they get exacerbation of disease. That's for sure some underlying mechanism. Whether like the atopic dermatitis is an allergic disease, I'm not so sure. I mean, there are also conflicting data on IgE, sensitization, IgE levels. IgE, omalizumab, for example, they are not really convincing data that IgE blockade really helps in general in atopic dermatitis. There might be, of course, subsets that might profit from it or profit at a certain amount, like reduce 30% of the activity. But this clear IgE functionality that you have in rhinitis or in asthma, this clear allergen trigger that might not be so universal in atopic dermatitis. And it all those descriptions on autoantigens that you find with IgE, it might all, we don't really know whether it's, I mean, there's so much inflammation going on, so much strong inflammation. And when you compare to psoriasis, the time that you suffer from this disease is so much longer. Even, I mean, you suffer from it from early life on. So there might be a lot of bystander inflammation going on that, get, I don't know, antigenic spread or so. So I think we don't really know it, but I don't know any convincing data that shows that really an antigen is driving the disease.
Sure. No, thanks for your insight. That's really interesting. Another really interesting question we have here is, how would you expect JAK or JAK1 specific blockade to be different from Dupilumab in terms of its molecular effect, and in particular on the 1 year of treatment samples? Would there be any difference there?
No, but we are eager to find out, of course.
Sure.
Given the resilience of those cells, my prediction is that you can't get rid of them with any treatment because those cells come back. There might be some nuance of the inflammatory profile, but of course that needs to be demonstrated. And we're of course very interested in that. But I would think you can't get rid of those cells also with JAK inhibition. Unless future studies will show that you get long-term disease remission, like for this data that you have now in psoriasis, when you block IL-17A and F, you get a long time of remission. I mean, so I think the clinical data will guide whether there might be some influence on those cells.
Sure, makes sense. Thank you. Okay, we have another question here. What do you think about the increase in Vegf signaling at the 1 year time point for Dupilumab?
Sorry, I didn't get the question.
Oh, sorry about that. So what do you think about the increase in Vegf signaling after a year of Dupilumab therapy?
You mean like on a protein level or on a transcriptomic level?
Yeah, the questions from the chat didn't specify. I suppose either.
So I'm not entirely sure whether increases in certain growth factors or chemokines, if you don't get like an anatomically correlate, whether there's any relevance in it. I mean, when you look at psoriasis, you get those tortuated vessels that normalize upon treatment. I mean, I don't see so much vascular differences in 1 year Dupilumab treated patients. So I'm not sure that really has a role in the one after Dupilumab treatment.
Sure. Okay. Great. Thanks. Let's see. I think that's it for the questions in the chat. Maybe we'll give it one more second in case anyone else comes in with any last minute questions. No, doesn't look like it. Yeah, thank you so much for your talk today. It's just really fascinating, and it's really neat to see using the latest techniques such as single cell RNA sequencing to really push this forward. It's just really exciting. And we've also used O-Link as well for some of our studies, and we really like them. So it's neat to see it working well for you, and appreciate the comparison of the suction blister fluid versus the biopsy data. I thought that was really interesting too. So thank you so much for joining us today. We really appreciate it.
Thank you so much for having me. Thank you. Thanks.
Great. All right, everyone, have a good day.